Today, I am talking about the basic cell structure and function in human cells readily in the cytoplasm. The parts, which are the nucleus, plasma membrane, and cytoplasm, they serve a specific function within the organism. The cytoplasm has cytoplasmic organelles, cytoplasmic inclusions, and cytosol, which is enclosed by cell membrane. The cytosol contains a liquid which acts as a site of multiple cell processes. It provides structural support and contains all soluble materials such as electrolytes and soluble enzymes. Golgi Apparatus located near the nucleus, which is composed of cisternae. It functions as sort, package, modify, and secretes the protein out of the cell. The lysosome is a spherical membrane-bound organelle that contains hydrolysis enzyme that functions to digest substance that enter the cells. Digest worn-out organelles involved in autolysis and extracellular digestion. The mitochondrion consists of outer and inner membrane. It has cristae on ribosomes and its own DNA. The function of mitochondrion includes ATP production, ammonia detoxification, apoptosis, production of heat, and more. The centrioles, also the organelles that present in eukaryote but not prokaryote. It main functions for cell reproduction. They will be long, stiff microtubules that split the cell apart during cell division. These are the differences between centriole and centrosome. These are their major differences in number of unit, arrangement, and condition in cells. Another important organelle is cilia, which is hair-like organelles that extend to the surface of mammalian cells. They are motile cilia and primary cilia. These are the description between both cilia, but the major differentiating factors is that motile cilia are used for moving, while primary cilia is used as antenna or receptor rather than movement. Next, flagella is a hair-like structure that emerges from cell wall and cell membrane. They consist of basal body, hook, and filament. It is mainly used by bacteria for movement. Our body, such as sperm, contain flagella for movement. After that, peroxisome is also an important organelle used for breaking down toxic materials in cell. It functions the same as lysosome, but mainly involved in digestion of alcohol, fatty acids, and amino acids. Next, ribosome, also one of the major organelles found in cytosol, and it has two subunits. It is used for the synthesis of protein, where to translate the specific protein that wants to be made via translation process. Ribosome can be free or located at rough endoplasmic reticulum. These are the mechanisms of both ribosomes in cytoplasm. Endoplasmic Reticulum, also an organelle that can be classified into rough ER and smooth ER. Rough ER studied with ribosomes that alter surface and mainly function for synthesis of protein, glycoprotein, and phospholipids. Smooth ER extend from rough ER to form network of membrane tubules. It functions mainly in fatty acid and steroid synthesis as well as drug detoxifiers. It also stores and releases calcium ions for muscle contraction.
The endomembrane system is a collection of membrane-based organelles that work together to create, modify, and export cell products such as proteins and lipids. It consists of the nuclear envelope, the rough and smooth endoplasmic reticulum, the Golgi Apparatus, and several types of vesicles. Let's start our tour with the endoplasmic reticulum. There are two types of endoplasmic reticulum, rough and smooth. The rough endoplasmic reticulum, or rough ER, is studded with ribosomes. These ribosomes are what give the rough ER its rough appearance. Ribosomes are responsible for producing proteins, which are packaged in the transport vesicles and carried to the Golgi apparatus. The smooth endoplasmic reticulum, or smooth ER, lacks ribosomes on its surface. This is why it's called smooth. The smooth ER is mainly responsible for the detoxification of harmful chemicals. It's also responsible for the production of lipids, which are also packaged in the transport vesicles and sent to the Golgi apparatus. The Golgi apparatus is made of a series of flattened sacs that look a little like pancakes. Its job is to package and distribute cell products that are either destined for a specific location within the cell, such as the plasma membrane, or for secretion from the cell. You can think of the Golgi apparatus as the post office of the cell, where packages are dropped off and destinations are assigned. For example, some of the proteins from the rough ER are modified by the Golgi apparatus to become digestive enzymes, which are packaged into a special type of vesicle called a lysosome. The job of this particular lysosome is to fuse with another vesicle coming in from the plasma membrane. The lysosome's digestive enzymes then break down the contents of the vesicle, releasing nutrients for the cell. Other cell materials leaving the Golgi apparatus may be destined for secretion from the cell. For example, some might be hormones that help with body regulation, while others might be proteins that become part of the materials surrounding the cell. Before these products can be exported from the cell, they must first be packaged into a secretory vesicle. This vesicle then merges with the plasma membrane, releasing its contents into the external environment. The endomembrane system can be modified slightly to meet the specific needs of each cell. For example, a muscle cell usually has more rough ER because of its need for protein, while a liver cell usually contains more smooth ER because of its role in detoxification. Regardless of these modifications, the movement of materials within the endomembrane system is basically the same for all eukaryotic cells.